Welcome back to our channel, BioLifer, and please click that red button below to subscribe for more great videos coming your way. Go ahead and like this video so that we can deliver more quality content to you guys. If you're new to our channel, don't worry, we can't wait for you to join the Bio family. And we hope you enjoy this video.
Hey everybody, it's Brianna, and today Joanna and I will be discussing the different organelles in a cell. But before we get into it, let's review what we learned about eukaryotic and prokaryotic cells. The main difference between eukaryotic cells and prokaryotic cells are that eukaryotic cells have membrane-bound organelles, while prokaryote cells lack them. Membrane-bound organelles are organelles protected by a single or double plasma membrane. Mitochondria, lysosomes, the endoplasmic reticulum, and the Golgi apparatus are examples of membrane-bound organelles. Because prokaryotes do not have a nucleus, the processes of transcription and translation of DNA replication can occur simultaneously while they occur separately in eukaryotes. Along with that, prokaryotes only have one copy of each gene, while eukaryotes have two copies of each gene. In turn, this explains why eukaryotes are able to replicate through the process of mitosis and meiosis when reproducing, while prokaryotes use the process of binary fission. Prokaryotes usually only have circular DNA called plasmids, cytosol, ribosomes, and a plasma membrane.
Hey guys, it's Kia again. Now let's discuss the two types of eukaryotic cells, which are animal cells and plant cells. Animal cells differ from plant cells in that plant cells have a cell wall made of cellulose, a protein, chloroplasts, which is where photosynthesis occurs, and a central vacuole, which stores water and other essential nutrients. Details of photosynthesis will be discussed in our next video. On the other hand, animal cells contain centrosomes and lysosomes, which plant cells don't have. Centrosomes contain centrioles from which spindle fibers develop in cell division, like for mitosis and meiosis, which you will learn soon. Lysosomes are organelles in the cytoplasm that contain digestive enzymes that break down waste products, worn down organelles, and digest viruses. Just for clarification, two centrioles make up one centrosome.
Plant and animal cells have a lot of similarities though. They both contain a nucleus, ribosomes, peroxisomes, mitochondria, smooth and rough endoplasmic reticulum, and the Golgi apparatus.
The nucleus of a eukaryote is like the control station of any operation. It coordinates the cell's activities, which include growth, intermediary metabolism, protein synthesis, and reproduction, which is cell division. In the nucleus of a eukaryote, it's important to remember that the DNA it stores is double-stranded. The DNA is made up of combinations of 4 nucleotide bases, which are adenine, cytosine, thymine, and guanine. Adenine pairs with thymine, while cytosine can only pair with guanine. A fun fact about DNA is that a human genome contains 3 billion base pairs of DNA, and DNA is shaped like a double helix. That's so cool, right?
The nucleus contains DNA, which stores hereditary information and regulates gene expression. So, it's basically described as being the control center of the cell. Ribosomes made of proteins and RNA bind to mRNA and tRNA to synthesize proteins, which is really important because proteins make up a majority of our body. Your body uses proteins to build and repair tissues. You also use proteins to make enzymes, hormones, and other body chemicals. Protein is an important building block of bones, muscles, skin, and blood, according to WebMD.
Serving two functions, peroxisome breaks down fatty acids and amino acids to be used to fuel respiration and form membranes as seen in the drawing. They also transfer hydrogen from compounds to oxygen in order to create hydrogen peroxide that can be converted into water. In liver and kidney cells, they work to break down toxic substances. Mitochondria is essentially the powerhouse of the cell because it carries out part of cellular respiration, which breaks down carbohydrates, fats, and proteins into usable energy. The products of glycolysis enter the mitochondria to continue cellular respiration. The Krebs cycle takes place within the matrix of the mitochondria, which is the gel-like material between the inner and outer membrane. The compartments are critical for the electron transport chain structure and function.
Today, I will be talking about the endomembrane system, which includes the endoplasmic reticulum, vesicles, the Golgi body, and the plasma membrane. There are two types of endoplasmic reticulum. There's a rough one and a smooth one. The rough endoplasmic reticulum has ribosomes on it, which has the ability to synthesize proteins. The smooth endoplasmic reticulum has the ability to synthesize lipids and steroids and also detoxifies the cells. In this example, we will be looking at the rough endoplasmic reticulum. In the rough endoplasmic reticulum, the proteins are modified, and once they are modified, they are formed inside a vesicle, and the vesicle moves towards the Golgi body and fuses with it, so the contents are dumped inside. Once the contents inside are further modified and packaged, two things can happen. The vesicle can form and turn into a lysosome, or a vesicle can form, move towards the plasma membrane, fuse with it, and dump its contents outside into the extracellular fluid.
We hope that after you finish watching this video, you will be able to name the organelles and their functions without any confusion. Thanks for watching, and we'll see y'all later. Bye!